Hello everybody, this is Dr. Kalpana V.N., Assistant Professor from the Department of Microbiology, Auxilium College, Vellore. I'm here to explain about nanobiotechnology, a new approach to treat and prevent malaria. Mosquitoes are medically important arthropod vectors, and it is one of the most important dangerous human health pests. Mosquito-borne diseases like malaria, dengue, West Nile, chikungunya, yellow fever, and Japanese encephalitis remains a major source of illness and causes death worldwide, particularly in tropical and subtropical countries. Mosquitoes alone were responsible for transmitting diseases to more than 700 million people annually. Among these diseases, malaria is a life-threatening disease which is caused by a parasite belonging to the genus Plasmodium that are transmitted to the people through the bite of infected female Anopheles mosquitoes. Malaria is the fifth cause of death among infectious disease worldwide, and it is the second cause of death in Africa, which comes after HIV or AIDS. It is a disease which can be treated within 48 hours, but it can cause fatal complications if the diagnosis and the treatment gets delayed. Despite centuries of efforts, malaria continues to infect millions and kills thousands of people. Next comes the World Malaria Report 2020, which is given by World Health Organization. There were 251 million malarial cases in 2010, to 19 million malarial cases in 2015, to 27 million malarial cases in 2016, to 29 million malarial cases in 2017, to 31 million malarial cases in 2018, and to 41 million malarial cases in 2020. Though there were 10 million fewer malarial cases in 2020 than when you compare with 2010, the data for the period between 15 to 20 highlights that there is no significant progress in reducing the global malarial cases during this time frame. Next comes the malarial status in India. On 4th December, the World Health Organization released the World Malarial Report 2021. So they said that there were 241 million malarial cases in 2020, which increased from 227 million cases in 2019. India alone accounts for 83% of malarial cases. Experts say the difficulties and the danger is still high. India carries nearly 2% of the global malarial burden and contributes to 87% of total malarial cases in Southeast Asia. Diagnosis and treatment of falciparum malarial cases continues to be a challenge for healthcare providers. To overcome these hurdles, innovative solutions along with the existing tool and strategies involving vector control, mass drug administration, and disease surveillance hold the key to solve this gigantic health problem. The Government of India has set a target to end malaria by the year 2030. Now comes the life cycle of malaria. The natural history of malaria involves the cyclical infection of humans and female Anopheles mosquitoes. In humans, the parasite grow and multiply first in the liver cells, then in the red cells of the blood. In the blood, there will be successive broods of parasite which grow inside the red cells and starts destroying them, thereby releasing a daughter parasite called merozoite, which continues the cycle by invading other red blood cells. The blood stage parasites are those that cause the symptoms of malaria. When certain forms of blood stage parasite are ingested during the blood feeding by a female Anopheles mosquito, they mate in the gut of the mosquito and they begin a cycle of growth and multiplication in the mosquito. So after 10 to 18 days, a form of a parasite called sporozoid migrates to the mosquito's salivary gland. When the Anopheles mosquito takes up a blood meal on other human, the anticoagulant saliva is injected together with the sporozoid which migrate to the liver, thereby beginning a new cycle. Thus, the infected mosquito carries the disease from one human to other, while infected humans transmit the parasite to the mosquito. In contrast to human host, the mosquito vector does not suffer from the presence of the parasite. As a result, the person exhibits three stages of symptoms like hot stage, cold stage, and sweating stage. In case of hot stage, there will be an intense heat, dry burning skin, throbbing headache, which lasts for nearly 2 to 6 hours. Then comes the cold stage, where the person feels intense cold, vigorous shivering, which lasts for about 15 to 60 minutes. The last stage is about sweating stage. There, the person exhibit a profuse sweating, the temperature gets declined, the person gets exhausted and weak, which lasts for nearly 2 to 4 hours. The majority of those who die from malaria are infants, children, and pregnant women. So there are four types of parasites which exhibit: Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. There are some drugs which is used for malaria treatment, namely chloroquine phosphate, artemisinin, mefloquine, and pyrimethamine. Each and every drug has its own advantages and drawbacks. So if you take the target of chloroquine phosphate, it mainly concentrates on heme metabolism. The main advantage of chloroquine phosphate is there will be a fast action in RBC stages, good bioavailability, a high volume of distribution. If you see the drawback, there is a widespread of chloroquine resistance. Macular retinopathy will be the drawback. And in case of artemisinin, it inhibits the Plasmodium falciparum ATP6 outside the parasite food vacuole. And in case of advantages, it is very safe and well tolerated. In case of the drawback, it is neurotoxicity dose dependent. Then comes the mefloquine. This targets heme metabolism, and the main advantage is the potent action in RBC stages, whereas the drawback is about higher rate of severe neuropsychiatric reactions and a very long half-life is there. In case of pyrimethamine, it mainly targets on dihydrofolate reductase inhibitor, and main advantages about pregnenolone and chloroplarnol, short elimination half-lives, and case of drawback, the metabolism varies considerably. Recently, it has been noted that the first vaccine against Plasmodium falciparum malaria, that is RTS-SA01, it is commonly known as Muscurix, showed only partial protection, that too particularly in infants. So there is a great increase in incidence of malaria mortality, that is the Plasmodium falciparum becoming resistant to commonly used antimalarial drug, and the Anopheles mosquitoes is becoming resistant to insecticides. Hence, the malaria becomes a very tough disease to tackle. Increased drug resistance, vector management, and vaccine development are all challenging. Therefore, to control the plasmodium as well as the vector activity, an inexpensive, eco-friendly, and effective antimalarial drugs are urgently required to eliminate this problem from every corner of the world. Hence, the major research area was said to be the nanobiotechnology. The advancement of green nanotechnology will bring a solution for both parasite as well as the vector control. So a green synthesized nanoparticles may be the right hop source for mosquito control agent. The use of green process for the synthesis of nanoparticle is a new and rapidly developing branch of nanotechnology. Now let's see about the nanotechnological approaches involved in treating the disease malaria. Current anti-malarial therapy mainly depends on chemotherapy. Therefore, it comprises serious toxic adverse events and development of drug-resistant parasites, which leads to treatment failure. To overcome the clinical failures involved in anti-malarial chemotherapy, a new drug development is of an immediate need. Nevertheless, the drug discovery and development process is expensive and is time-consuming. Globally, several universities, research institutions, pharmaceutical industries, and a non-profit organization like Medicines for Malaria Venture, they have designed and synthesized a novel anti-malarial compound and tested against Plasmodium parasites in both in vitro and in vivo models. However, these have shown some extent of toxicity in the preclinical studies. The complexity of parasite life cycle, scattered the locations, that is erythrocytic and exoerythrocytic stages of malarial parasite in the host. An incomplete knowledge on biology and pathology of malaria parasite has created a very great challenge for the researcher to develop a new drug against malaria. Recently, nanotechnological approaches have gained an importance for target-specific drug delivery of drugs with improved safety and efficacy. The application of nanotechnologies to medicine or nanomedicine is a field of science having extensive applications and a great impact on biotechnological and pharmaceutical industries in designing and developing a nanostructure material which is encapsulated with antimicrobial agents, acts as a promising approach to encounter the complication as associated with existing antimicrobials. The primary goal of nanotechnology-based malaria therapy is to deliver an anti-malarial chemotherapeutic agent which is encapsulated in nanomaterial to target the parasite-infected erythrocytes and intracellular parasitic vacuoles. Additionally, the use of nanomaterials will permit augmented efficacy, safety, selectivity, which alter the drug pharmacokinetic properties, improves the solubility profile of drug, avert drug degradation, and endorse a constant release of drug directly at the target location. Encapsulation of more than one chemotherapeutic drug with nanovector allows the combinatorial treatment which have a synergistic effect. Numerous nanoparticles like liposomes, solid lipid nanoparticles, nanostructured lipid carriers, metallic nanoparticles, nanoemulsions, and polymeric nanoparticles have been explored and found to be very advantageous for drug delivery applications as nanocarriers. Hence, these nanotechnological based products may attest to exceed the limitations of existing therapy with respect to safety, efficacy, and cost effectiveness, which improve the patient's compliance to treatment. Recently, United States Food and Drug Administration approved liposomal amphotericin B for the treatment of visceral leishmaniasis, a neglected tropical protozoal parasitic infection, and they have shown an improved efficacy and safety. The surface modification of nanoparticles which encapsulated antimalarials will increase the selectivity and specificity against target, which results in improved safety and efficacy. Several research groups have designed and formulated a nano-based products with standard anti-malarial drug, and they have screened against plasmodium parasites infected against animal models, and they have shown promising results, but there should be a greater attentiveness of the scientific communities to such a poverty-stricken tropical infectious diseases. Now comes the green nanotechnology prospect to control malaria. Green nanotechnology integrates the principles of green chemistry and green engineering to formulate safe and eco-friendly nanocomplexes to fight against impediments affecting the human health or environment. Consequently, a combination of green nanotechnology with drug delivery area has essentially initiated a unique realm of green nanomedicine. For example, we have a silver nanoparticle which is synthesized by green nanotechnology using Andrographis paniculata, which shows an antiplasmodial effect against Plasmodium falciparum. Eco-friendly and biosynthetic silver nanoparticles prepared from Ashoka and neem leaf extracts also show the ability to inhibit the parasite effectively. Green nanotechnology-based zinc oxide nanoparticles, which are synthesized by using aqueous peel extract of Lagenaria cicharea, that is a common bottle gourd, also showed effective against Plasmodium falciparum. These zinc oxide nanoformulations inhibited the formation of hemozoin, which is required for the parasite to survive. Emergence of drug-resistant parasite, severe toxic adverse events of existing anti-malarial chemotherapeutics, and non-availability of proper vector control measures and vaccines against malaria leads to a very difficulty in malaria control and treatment. Malaria is one of the most prevalent and serious public health issues of tropical disease worldwide, as we all know. Hence, many research organizations were trying to develop a therapeutic strategy to combat malaria, but no satisfactory results were obtained. The expansion of nanotechnological based therapeutic agents against malaria has endangered excessive interest in formulation and drug development among scientists. Nano-based drug delivery systems have shown exciting data in preclinical studies, thus demonstrating their potential as therapeutic carriers. Various nanoparticles like liposomes, solid lipid nanoparticles, nanostructured lipid carriers, metallic nanoparticles, nanoemulsions, and polymeric nanoparticles have been explored as a drug carrier system for antimalarial chemotherapeutic agents to target malaria with improved efficacy and safety. Various research organizations have designed and developed nanotechnology-based formulation with standard antimalarial drugs. Some of these nano-based drugs have been preclinically studied and shown a positive outcome. Nonetheless, a superior attention of the scientific communities is crucial for such a potentially life-threatening infectious diseases. To conclude, significance of expected outcome in relation to public health. The outcome of this nanotechnology study will be a breakthrough in the area of malaria research. It may be a strong alternative to current anti-malarial drugs and drug combinations to combat with the drug-resistant parasite. Although it is premature to conclude at this stage that these herbal and microbe-based nano combinations can be used as effective antimalarials, this finding will provide a foundation for further exploration of new effective herbal compounds with nanoparticles for protection from malarial parasites. Thus, the present nanotechnology-based study will benefit the society by reducing the morbidity and mortality which is caused by malaria. For further doubts, clarifications and queries, please go through my publications. I raise my hand to defeat malaria. Zero malaria starts with me. Thank you one and all for your patience listening.